Next, good morning students. Next, here that it is we were discussing about the pancreatic enzymes. Okay. With pancreatic enzymes. Here, one of the most important enzyme among the pancreas, which is secreted by the pancreas, that it is the trypsin. What it is? It is the trypsin. Here, the trypsin is secreted in the form of inactive, or it is secreted in an inactive form. Which form? That it is inactive form. Okay. So, what is the inactive form here? Means that it is the trypsinogen. What it is? That it is the trypsinogen. Here, the trypsinogen gets converted into the trypsin in the presence of an enzyme, and the name of that enzyme is the enterokinase. What it is? That is the enterokinase. This enterokinase enzyme is secreted from the intestinal tubes. Okay. So here and here there is an involvement of some hydrogen ions, due to which we can call this reaction as the autocatalytic reaction. Okay. So next thing, trypsin will act on the proteins. Here, this trypsin acts on the proteins. Trypsin acts on proteins. Okay, which will act on the proteins and converts into the polypeptides and amino acids. Trypsin gets converted into polypeptide and amino acid. So let's see the reaction, how this trypsin is going to convert the protein into polypeptides and amino acids. So what are that? Means here the protein, this is the first reaction of the pancreatic by the pancreas. The second reaction is that the protein in the presence of an enzyme trypsin, and what is the pH over here is that it is 7 to 9, alkaline pH. Okay, it gets converted into polypeptide and one more amino acids. This is the second reaction of pancreatic enzyme. So here, how this protein is not going to directly convert into polypeptide and amino acid. And how it is going to convert means, there are certain substances. First, this protein is going to convert into that one, then finally to the polypeptide. So what is that means? First, protein will convert into alkaline metaproteins. Proteins first, it gets converted into alkaline metaproteins. Okay, alkaline metaproteins. The next into the primary proteoses. Then into the secondary proteoses. Then into the secondary, then into the peptones, and lastly into the polypeptides. Lastly into the polypeptides. So here trypsin First, it will convert into the first of all, the trypsin is secreted in an inactive form, that is the trypsinogen. Here you see, this is the inactive form. It gets converted into active form. What is that? That is the trypsin. Okay, by which enzyme? Enterokinase. The next thing here, protein will convert into the polypeptide and amine. What is the pH over here? It is 7 to 9. The next thing, directly it is not going to convert directly into the polypeptide and amino acid. First into alkaline metaproteins, primary proteases, secondary proteases, then peptones, then polypeptides. Okay, so this is about the trypsin. And second enzyme is a chymotrypsin. What is the second enzyme? It is the chymotrypsin. Let's see about that one. Next one, that it is chymotrypsinogen. Second one is chymotrypsin. This is the second enzyme. So let's see here. This chymotrypsin, it is also secreted in an inactive form. What is that inactive form of chymotrypsin? That is the chymotrypsinogen. What it is? This enzyme also secreted in an inactive form. And what is the inactive form? That is the chymotrypsinogen. What it is? That it is the chymotrypsinogen. So here, how it is going to convert means, which is an enzyme which is going to convert from inactive to active form. Let's see what is the reaction. Chymotrypsinogen converted into the chymotrypsin in the presence of an enzyme, that is the trypsin. Which enzyme is responsible for conversion of this inactive into the active form? That is the trypsin at pH 7 to 8. Here chymotrypsinogen is an inactive form. It gets converted into active form. Okay, so this is about the chymotrypsinogen. Here trypsin hydrolyzes peptide linkage. Next point about the trypsin. Trypsin hydrolyzes the peptide linkages, and these peptide linkages involve the carboxyl groups. It involves the carboxyl Again, this is an essential point of trypsin, where it hydrolyzes the peptide linkage, and that peptide linkage will have the carboxyl groups. groups of what? Of amino acids. Okay, carboxyl groups of amino acid. And what are that amino acid? Arginine and lysine. Carboxyl groups of two amino acids are there. That is arginine and one more, that is lysine. Okay, these are the two amino acids. So next comes the third enzyme, that it is the carboxypeptidase. Let's see about that one. Next comes the carboxypeptidase. Third enzyme, carboxypeptidase. Okay, here it is activated both. This is also will be in the form of inactive form. Okay, this is gets activated into both, that it is trypsin and enterokinase. Here carboxypeptidase hydrolyzes polypeptides to simple peptides. It hydrolyzes carboxypeptidase into polypeptides and peptides. This is the first action. And one more thing, this enzyme is also secreted in an inactive form, which gets activated by two enzymes. What are that? That is a trypsin and one more, that is enterokinase. These both enzymes are responsible for activating this carboxypeptidase. Okay, the next thing here, that simple peptides, polypeptides then into the simple peptides. These peptides will be in the form of simple peptides. Okay, and here simple peptides as well as the amino acids, polypeptides, simple peptides and amino acids. These three are the products after the hydrolysis of carboxypeptidases. Okay, so the next thing, this enzyme, there are two carboxypeptidases, that is A and B. What are that? Two carboxypeptidases, that is carboxypeptidase A and one more, that is carboxypeptidase B. This one. There are two types. Okay, here carboxypeptidase act on the peptides having a free carboxyl group on a terminal aromatic amino acid. Here you see A acts on the peptides having a free, acts on the peptides having a free carboxyl group, having a free alpha carboxyl group, on the terminal aromatic amino acid. On the terminal aromatic amino acid. The next one here, carboxypeptidase B is there. This carboxypeptidase B acts on the peptidase having alpha carboxyl group on a terminal basic amino acid, not aromatic, on the terminal basic amino acid. Acts on the peptides having a free carboxyl group on the terminal. Same thing acts on the peptides having a free alpha carboxyl group, which is terminal basic amino acid. Okay, so this is about what? About the carboxypeptidases. Next come the what is the reaction of this carboxypeptidase? Means here polypeptides gets converted into the simple peptides and amino acids. Simple peptides and amino acids in the presence of an enzyme, that is the carboxypeptidase. Okay, altogether we can say polypeptides gets converted into simple peptides and amino acids in the presence of an enzyme carboxypeptidase. Okay, next enzyme is the pancreatic lipase and one more, that it is the pancreatic amylase. Let's see about that one. Next enzyme is the pancreatic lipase. Pancreatic lipase. Okay, here the pancreatic juice contains a lipase, and formally we can call it as the stearin, which hydrolyzes fats into glycerol and fatty acid. Here, pancreatic lipase or pancreatic juice contains a lipase. Okay, that lipase enzyme we can call it as the pancreatic lipase, which is also called as the stearin. What it is? Stearin. So this enzyme hydrolyzes fats into glycerols, and fatty acids. Okay, here pancreatic lipase hydrolyzes fats into glycerol and fatty acids. Okay, its activity is accelerated by the presence of bile salts. Whatever this reaction is going to take place, it is accelerated by What means that is by the bile salts. Bile salts will be present in the bile juice. The bile juice which is secreting from the liver. Further we will talk about that one. So pancreatic lipase is by far the most important enzyme concerned with the fat digestion. It is one of the very important enzyme for the fat digestion. Okay, the next comes the pancreatic amylase. Fifth enzyme, that is the pancreatic amylase. Okay, here the pancreatic amylase, it is similar to the salivary amylase. A starch splitting enzyme is found in the pancreatic juice. Pancreatic amylase, it is a starch splitting enzyme what it is? It is a starch splitting enzyme found in the pancreatic juice. It is found in the pancreatic juice. Okay, the next here. So this pancreatic amylase hydrolyzes starch to maltose. Here it is hydrolyzes starch to maltose, or starch gets converted into maltose in the presence of an enzyme pancreatic amylase. Okay, the next thing here. Last enzyme is the pancreatic nucleuses. Sixth one, that it is pancreatic nucleuses. Okay, here the pancreatic nucleuses, 2 polynucleotides occur in the pancreatic juice. Here, the 2 polynucleotides occur in the pancreatic juice. And what are the 2 polynucleotides? So, they are the unhydrolyzing RNA and DNA. RNA and DNA. So, this is about what? About the pancreatic enzymes. So, whatever the regulation of pancreatic juice is there, it is under the control of 2 phases. One is nervous phase and the another is the hormonal phase. In the nervous phase, whatever the gastric juices are going to secrete, that is nerve impulses arising from the stimulation through the sight, smell or taste of food reaches the pancreas through the branches of vagus nerves. Okay, from that side of smell of food, taste and all, from there it is going to transfer to the vagus nerve and the pancreatic juices starts to secrete. This is about the control by the nervous system. One more is there, that is by the hormonal action. Nitric acid, that is chyme, from the stomach enters into the what we call small intestine. The cells, it stimulates a hormone, secretion of hormone, and that hormone is known as the secretin. Here the secretin is absorbed into the blood and carried by the blood to the pancreas. Okay, which stimulates the secretion of, what we call, that is the stimulation of secretion of secretin. Okay, so this is about the pancreatic juice. Next, we'll go for the next topic, that is the bile. Next term is bile. Okay. Here, in addition to pancreatic and intestinal secretion, a third secretion poured into the duodenum. See, yesterday also I told you, this is the esophagus, this is the stomach. Okay, this is the stomach. Here there is a presence of pancreas. Okay, then here a largest gland will be there, that is the liver. This one is the liver and this is the pancreas. Here there is a presence of a gallbladder. All the ducts from the liver, it will communicate with the pancreatic duct, and then both ducts are going to open into a common passage and a common duct that is known as the hepatopancreatic duct. What is hepatopancreatic duct? Okay, so here this secretion from the liver, what is that secretion means? That is the bile juice. This plays a very important role in the digestion. Okay, this secretion is going to take place. This is the U-shaped or the first part of the small intestine, to which we can call it as the duodenum. What it is? That it is the duodenum. So here, although there is no action of the bile juice is going to, see, it is going to stimulate the pancreatic juice as well as the intestinal juice. So the bile juice which is produced from the liver and is poured into the intestine through the duct, that this duct we can call it as the bile duct. And what is this small pouch-like structure is known as the gallbladder. So this gallbladder, what is the role of gallbladder? Means here, whatever the bile juice is secreting from the liver, whenever there is no need of the bile juice, at that time this gallbladder will store the bile juice. Okay, whenever it is required, the gallbladder will stimulate the ducts, that is the bile duct, to release the pancreas. That is the bile juice. So here, this secretion of bile juice, it is also stimulated by an enzyme, and the name of that hormone, that is the hormone. See here, bile juice secretion is stimulated by an hormone. And what is the name of that hormone is? It is secretin. What it is? It is the secretin. Okay. The next, what is the composition of bile? See, before going to composition of bile, here the pancreas are also going to stimulate the secretion of bile from the liver, and the gallbladder is this gallbladder is under the control of an hormone, and that hormone is known as the polycytokinin hormone. The name of that hormone is the polycytokinin. Okay, so this is about the two hormones, that is secretin and one more polycytokinin. Secretin will stimulate the secretion of bile juice from the liver, and polycytokinin, which is going to stimulate the secretion from the gallbladder. Okay, the next here the bile. What is bile means? It is one of the viscous liquid, alkaline in reaction and bitter in taste. So having a color from yellowish brown to green in color. This will be the color of the that is the bile juice, and it is what are the important constituents of this bile juice means, that is the bile pigments, bile salts and cholesterol. Okay, so this is about the bile juice. Next, in the next class we will see about the bile pigments and bile salts. Okay, thank you.
No, no. Just